and welcome to this course in basic pharmacovigilance. The Uppsala Monitoring Center, the UMC, works with patient safety and pharmacovigilance in a global context. As the WHO Collaborating Center for International Drug Monitoring, our main tasks are to perform research and signal detection on data in Vigibase, the WHO global database for individual case safety reports, and to provide support to the member countries of the WHO Programme for International Drug Monitoring. The UMC also develops tools, methods, and dictionaries to be used in the field of pharmacovigilance. And we offer courses and training material to countries to enable them to create their own national PV systems and thereby contribute to the global PV network. If you're interested in knowing more about the UMC, you're most welcome to visit our website for further information. Attached to this lesson, you will find a list of terms that are commonly used in pharmacovigilance. We recommend you to read through it before you watch the lessons in order to better understand the concepts that are being discussed, especially if you're new to the field of pharmacovigilance. Please note that some of the lessons also have additional material attached to them if you want to deepen your knowledge. We will guide you through the basic concepts of PV and show you examples of why and how pharmacovigilance plays an important role in improving patient safety. Let's begin.
Welcome to this lesson about the history of pharmacovigilance. Let me start off with a broader perspective on history in our particular context. How can we make use of history in practice of medicines and healthcare? Can the past offer lessons for the present and guide us for the future? Simply put, can history teach us what to do? Well, in this lesson, we will discuss a few examples from the past that have indeed guided development of what to do and what not to do in order to improve patient and medicine safety. The history of regulating drug and medicinal products stretches back long before the 20th century, and the development of regulation has generally taken place in a context shaped by opposing forces. On the one hand, market forces comprising manufacturing and trade agendas pushed hard to promote products and increase sales. On the other, advocates for public health pushed for better testing and monitoring of drugs. As a result, drug regulation to safeguard public health, improve efficacy and safety evolved in a stepwise rather than linear manner. Over time, harmful pharmaceutical events led to legislation and regulations that more clearly defined what constitutes prescription versus non-prescription drugs, introduced rules mandating formal licensing of products to be on the market, and controlled information and advertising of medicines and remedies directed to the public. Despite these advances, during the first half of the 1900s, there were still gaps evident in the testing of drugs for safety. This led to increased calls for a stronger body of scientific evidence, control testing to prove the efficacy of individual drugs, and the active monitoring of the whole life cycle benefits and risks of products already on the market. Some of the most considerable milestones took place during the 1900s, driven by political decisions or lack thereof. That period will therefore be of particular focus in this lesson. Let's start by looking at one of the worst pharmaceutical catastrophes in history, from which many difficult lessons were learned, namely the drug thalidomide. Thalidomide was a medication introduced in the late 1950s as a sedative, but marketed in several countries and widely used off-label for morning sickness in pregnant women. In some countries, it was even sold over the counter. In early 1961, Dr. William McBride, an Australian obstetrician who was among the first doctors to sound an alarm about thalidomide, delivered a baby who had malformed arms and other problems. The birth defects he noted are part of the condition called phocomelia, which resulted in short, seal-like limbs in these children. Children born with phocomelia also suffer from other complex malformations of inner organs, and many do not survive the immediate neonatal period. Within just a few weeks of his first observations, Dr. McBride delivered 2 more babies with similar malformations. In a letter published in the medical journal The Lancet in December 1961, he described his observations of an increase in birth defects that he suspected were attributable to the drug thalidomide. His letter explicitly asked the journal and its readers if the global community had experienced any similar events. Have any of your readers seen similar abnormalities in babies delivered by women who have taken this drug during pregnancy? And about the same time, a German doctor, Widukind Lenz, made the same connection and documented cases all over Germany. Following this, the drug was quickly banned or withdrawn from the market in one country after another. We can consider this to be one of the first recorded examples of how rare adverse events could be evaluated by using the collected knowledge and pooled information and data of the global community. Thalidomide was the biggest man-made medical disaster ever. More than 10,000 children were born with a range of severe debilitating malformations, along with an unknown number of fetal deaths. This catastrophe changed the course of medical history, driving many significant developments in the years that followed. The thalidomide scandal led to the beginning of both national and international collection of ICSRs to further benefit patients and improve medicine safety and decision-making among healthcare professionals. For example, in 1963, the UK's NHS set up a special committee to create a system to enable systematic reporting of adverse drug reactions, which became known as the Yellow Card Scheme. And in 1968, the WHO established its Program for International Drug Monitoring. Regulation also developed. In addition to issues of pharmaceutical quality, more emphasis was put on the safety of medicines. Furthermore, mandating proof of the efficacy of a drug and its safety before marketing also led to more rigorous drug approval and monitoring systems being put in place at some regulatory agencies in the countries such as the USA and the UK. Another example, one that contributed to the consideration of variances in subsets of patient populations during drug development, came 2 decades later with the drug benoxaprofen. This was a new anti-inflammatory drug marketed for treatment of arthritis in 1980. It was marketed massively to the public on radio and in newspapers, with advertisements encouraging patients to ask their doctors to prescribe it. However, the first report of deaths associated with benoxaprofen came 2 years after its introduction, with an increasing number of reports of elderly patients displaying cholestatic jaundice and sometimes fatal liver toxicity. It was identified that there were a number of risk factors predisposing patients to benoxaprofen-induced cholestasis, and fatal reactions occurred predominantly in the elderly. Later, it was also shown that the elimination of benoxaprofen in elderly patients was much greater than in healthy younger adults. This was one of the experiences that led the US FDA to issue a guideline requiring sponsors to study the pharmacokinetics of drugs in the elderly if the drug is likely to be used by this population. It was an event that also led to development of internationally harmonized guidelines on pharmacokinetic studies in drug development and better understanding of patient subpopulations. It made way for subsequent guidelines to recommend analysis in other subpopulations as well, such as in females, different ethnicities, and patients with hepatic or renal dysfunction, to study the potential for altered pharmacokinetics of drugs, and therefore the clinical response. During the 1990s, an emerging body of case reports were published in the scientific literature on drug-drug interactions that altered metabolism of medicines, resulting in toxic levels of drug metabolites. A number of non-cardiac drugs were first reported in isolated published case reports to prolong the QT interval of the electrocardiogram with or without torsade de pointe, which is a specific type of abnormal heart rhythm. And these findings were seen even in patients who were taking the recommended prescribed dose of their drug. Investigations gave more understanding of mechanisms behind this. With excessive levels of one parent drug, and a proportionate reduction in the concentrations of its metabolite led researchers to suspect an inhibition of the metabolism of the parent drug. This was later confirmed to be the case. Whereas the parent drug prolonged the QT interval of the ECG and was torsadogenic, the metabolite was a therapeutically active component lacking of this toxic effect. More shared reports of similar cases ultimately led to development of guidelines on drug interactions in the EU in 1997 and subsequently in the US in 1999. The collected findings made previously also led to additional recommendations and points to consider document to be adopted in the EU, which eventually evolved into 2 internationally harmonized guidelines. These guidelines require the sponsors of a clinical trial to, among other things, specifically investigate the potential effect of a drug, even on cardiac drugs, on cardiac functions such as the QT interval. In summary, as devastating as some of these drug catastrophes have been to the patients and their families, their suffering has not been in vain. They have provided us with learning experiences that have changed the pharmaceutical industry, guided the politics of drug development, and inspired the creation of systems to prevent harm from medicines and share early warnings of potential risks. In particular, these lessons have led to a shift in drug regulation to focus more on patient safety and efficacy rather than simply pharmaceutical and product quality, a better understanding of patient subpopulations and the need to consider such variances during drug development, and finally, a better understanding of the need for clinical trials designed to investigate, among other things, the potential effect of non-cardiac drugs on QT intervals. There are many more events and experiences that we have learned from that have shaped how the field of pharmacovigilance is practiced today. Pharmacovigilance is now defined as the science and activities relating to the detection, assessment, understanding, and prevention of adverse effects or any other drug-related problem. The science is being actively promoted and developed by WHO at the global level through its Program for International Drug Monitoring, and in a decentralized way by many other actors such as the officially appointed collaborating centers to the program, regulatory agencies, academia, and industry. Effective pharmacovigilance is key to enhancing patient care and patient safety in relation to the use of medicines. It also provides vital support to public health programs by providing reliable, balanced information for assessing the risk-benefit profile of medicines. Today, more than 130 countries are part of the WHO pharmacovigilance program, learning from past mistakes and actively seeking knowledge to prevent harms caused by medicines.